Thanks very much. It's a great pleasure for me to be here, to have a chance both to come back to Ann Arbor, which I visit with great pleasure, and also to talk in Alan's honor, because I have these two slides from Alan's visits to Boulder, and you can notice there's a glass in his hand at each point. But he didn't spend all his time with glasses in his hands. He was a very active member of the lab and a wonderful member because of his curiosity, his intellectual acumen, and his experimental skill, and his self-confidence, which was really very impressive. I'm one of these people who's always a little unsure about whether I'm about to step on something bad and make a mistake. But Alan was just forging ahead. And he did a great job in my lab. But he went on, and when he came to Ann Arbor, he just took off as far as I was concerned, and did a truly remarkable job of innovating in several fields, including the kind of science that he did when he was visiting me, and which I've continued with, but also in other things having to do with instrumentation and engineering topics of a kind where his own mind, which was an excellent mind, was able to conceive of good questions, good ways to address them, and come up with important answers. He's a great scientist, I think, and we're going to miss not only him as a person, but the science he would have done if he'd lived longer. I've had a lot of colleagues in my work. Alan is there on the second row, as you can see, and was involved with some of the work we've done on mammalian cells. But what I will describe is almost all, of course, the work of other people, because as you get on, you tend to be a lab director rather than a lab scientist. I'm happy to say I've been retired now for 7 years, and I've had the pleasure of being able to maintain my grants, and now I'm a scientist again. So as I get to the end of my talk, some of the work will actually be my own, but most of it will be that of these fine people here, and I'll mention them as I go along. What I'm talking about is the process that's essential for cell division. And what you see here is a few mammalian cells that are growing in cell culture viewed by a microscope. And if you pick any one cell and watch it for a while, you'll see it divide. This is time-lapse cinematography. And this movie was actually taken 40 years ago in my lab. So if it looks a little grainy, it's because it's gone through a fair number of transductions. This is a wonderful way to describe the process that I've been interested in and that Alan was interested in for a lot of his life. In order for a cell to divide and produce two daughters who can go on to divide again, of course, they have to have chromosomes, because the chromosomes are the book of instructions which are necessary for building all the parts of the cell. So the segregation of the chromosomes is a fundamental event which must precede the actual cell division itself. So every cell here is in the process of going through metabolism to keep itself alive, but it's also doing biosynthesis in order to make enough ribosomes, enough membrane, enough muscle proteins, enough enzymes to be able to divide and then do it again. And then, of course, there's DNA synthesis that must occur. The entire cell cycle is occurring during this interphase period, and all this movement is these cells wandering around, feeding on their environment, taking in food, which of course will be both building blocks and energy in order to do the syntheses, the events that are required to have two cells that were identical to the one of the first. Now, this kind of growth is, of course, very important for many aspects of the way in which we develop from a single fertilized egg into the bodies that we know and we hope love. It also can go wrong, however, and these cells are actually cancer cells, and you can see now that they're dividing all over the place. This is not because they're dividing any faster, but there are just more of them. The cell cycle time is constant. But these cells don't know about regulation. So this phenomenon, which is so advantageous to us in so many ways, can accumulate cells where they don't belong, which is an important factor, of course, in Alan's cancer and in, I must say, the cancer that took the life of my oldest son when he was only 31. So we study cell division with a mixed sense of admiration for all the important things that it does, like maintaining the normal complement of blood cells. As you sit here wondering whether this lecture is going to get boring, your blood is being reproduced by the bone marrow And there are about 1 million cell divisions going on every second in your bone marrow alone in order to replace all of the red blood cells that are dying. And if they don't do it, you become anemic. So continuous cell division is important for renewing both inside and out, for repairing wounds, accomplishing procreation, of course. So it's no surprise that many people have studied cell division. In addition, of course, when cell division goes without control, as in cancer, it becomes a major medical problem, and so figuring out ways to downregulate cell division is at the heart of most of what are regarded as effective chemotherapies today. We hope that we'll find chemotherapies which are specific for cancer cells, but at the moment that's usually for only a subset of cancers, and the majority of chemotherapy that's done is simply to treat cells with poisons that are more effective on cells that are growing rapidly, such as DNA synthesis inhibitors or inhibitors of the machinery that brings about chromosome movement. So I've spent most of my career studying this machinery for chromosome movement, under the illusion that it's important and that I might actually do something that would help elucidate its mechanism. I'm going to show you a movie of that division now because it's really quite a remarkable process, and perhaps the most interesting thing you'll see in the whole talk. These are chromosomes. This is not a human cell. This is a cell from a newt. And it's chosen for this movie because newts have huge cells and very big chromosomes, but about the same number of chromosomes as you find in a human cell. This is the envelope around the nucleus. These are chromosomes after they have condensed. And if I now start the movie, what you'll see is the breakdown of this envelope. And the chromosomes then appear to, I'm sorry, that wasn't what we wanted. We wanted to get that little button down there. The chromosomes will now appear to start moving in ordered ways. Initially, the order is not so evident, but you'll see them gradually accumulate on a plane. This is the formation of a planar array in which every chromosome is attached to an object down here called a spindle pole and another object up here called a spindle pole. And this organization then is almost a twofold symmetric bipolar structure, and those chromosomes will now segregate, and one copy of each chromosome goes to each daughter cell. The cell pinches in the middle, and these two cells are now genetically equipped to be able to grow and divide again because they have a complete copy of the necessary set of chromosomes. Amazing. So how does it work? I saw a movie like this as a graduate student, and I became absolutely fascinated by the process, and I've been trying to help understand how it works ever since. Here you can see the DNA stained blue, and in the cytoplasm are microtubules, that is, polymers of the protein tubulin, and these are cells which were prepared for microscopy at different stages in the mitotic process. And you can see that when the chromosomes are aligned in that stage where they took on order, and I referred to the structure as twofold symmetric, it really is. This red structure is the mitotic spindle, named because it was named at a time when people did still spin wool, and it reminded the person who first saw this morphology of a spindle for spinning yarn out of wool from a sheep. So the mitotic spindle has the ability to push all the chromosomes into this planar array at the equator of the spindle and then separate them and elongate so that when that cleavage furrow that you saw comes in, we now have two separate pools of DNA ready to allow the cells to go through another cell cycle, as you saw in the first time-lapse movie. The spindle depends for its accuracy and function on microtubules, shown here in a classic electron micrograph taken by Robley Williams Sr. with protein purified by Mark Kirshner. In the background is the soluble protein, tubulin. This is a microtubule which forms by the addition of these little subunits onto the end, and the polymer can elongate through that addition. When you depolymerize a microtubule, it depolymerizes into a lot of these little subunits, which are a protein of about 110 kilodaltons, and these rings, oligomers of tubulin, which are formed by taking these strands that you can see in the wall of the microtubule. Those are called protofilaments, and they peel back and form these sort of curled oligomers of tubulin. And I'll come back to this because I think it's important for the way in which chromosomes move. But this picture shows you the static states at either end of a dynamic equilibrium in which protein, under the right conditions, can polymerize to form microtubules or depolymerize to form the soluble protein. When tubules are forming in a mitotic spindle, that assembly is tightly regulated. This is an electron micrograph with the chromosomes shown as these big black objects in the middle. These are the spindle poles. The microtubules are shown here by staining them with gold. And you can see there are astral rays of microtubules projecting outwards, bundles of microtubules projecting inwards towards the chromosomes, and they look as if they attach at specific sites. To understand the mechanism for this complex process, we need to understand how is the assembly of microtubules controlled, and how do they attach to the chromosomes, and how do we build this twofold symmetric structure, and how does it function to pull the chromosomes apart? It becomes a series of questions about microtubules, their control of assembly, their attachments, and what do they do? This is the structure that you find at the poles of the cell. And at that pole is this fancy looking thing. And these structures, which are called centrioles, have attracted a huge amount of attention. But it turns out that what's really important for establishing a mitotic spindle is this rather unprepossessing fuzz around one of the centrioles, which is called pericentriolar material. And it has that polysyllabic name because nobody knows what it is. That's no longer quite true, but it's taken quite a long time to try to identify the proteins that are there. We now know that the key protein that's there is actually an unusual form of tubulin called gamma tubulin, which doesn't polymerize to make microtubules. It forms a little ring, and the ring is shaped like a lock washer with one side just above the other, and there are about 12 other proteins depending on the species from which you isolate this thing. And this little cup is an initiator for the growth of a microtubule. And the stuff in the pericentriolar material binds the gamma tubulin complex and therefore initiates the growth of microtubules that come out from it. And centrosomes have been isolated, not in pure form, but in form to study. And in this image here, which is a movie prepared by Tim Mitchison's laboratory, and you may have seen it because it's available as a teaching tool, you now can see the centrosome, and although this is time-lapse, this is actually what microtubules do. This is not sped up by more than about a factor of 4. And these microtubules grow and shrink, all initiated from the centrosome. And this event is going on inside a cell from that spindle pole, meaning that although the spindle looks as if it is a single structure, it's actually growth and shrinking of these microtubules as they reach out to interact with things. What they're interacting with, of course, is the chromosomes. And this image shows that a mitotic chromosome at the stage when it's attaching to the spindle has two parts, because this is one copy of the DNA and this is the other copy of the DNA, because the whole goal of this process is to have these two identical things be organized so that one part will go one way and the other part will go the other way, so that each of the two daughter cells will get a full copy of the genes. And this little fuzzy structure on the surface has been called the kinetochore because it is the specialization that develops on a chromosome that attaches to microtubules, binds them, interacts with them, and becomes both a motor and a signaling device in order to try to assure the accurate segregation of chromosomes. So understanding this junction becomes very important because you can imagine the centrosomes, which are at the poles, are initiating these microtubules that grow and shrink and grow and shrink. And if I'm a chromosome over here, I want to capture microtubules so that I can be drawn to the pole. But I also have the problem that my sister, who has her back to me and her kinetochore facing the opposite direction, she has to be able to interact also. And the key thing is that sister chromosomes have to attach to sister poles because we want to achieve a twofold division, And for that to happen, it must be that if this kinetochore connects to the west, this one must connect to the east. So that's the fundamental nature of setting up the mitotic spindle. And once it's been set up, of course, then you have the whole issue of how are you going to move things. And our ideas about how you're going to move things have grown in phases, really. One of the important phases in my own career was the discovery of microtubule-dependent motors. And when I was a graduate student, Ian Gibbons discovered dynein in cilia and flagella, and that was the first motor that was known. And dynein was an extremely important contribution, and indeed, we've been able to show that dynein is present in the spindle. There are multiple forms of dynein. The forms that are part of the cilia and flagella are not in the spindle, but you see these little 2 dots here and 2 dots there and 2 dots there. Those are the kinetochores of chromosomes, and they bind dynein, as shown here with immunostaining with a fluorescent antibody. It's not the only place that it is. It's also at the poles and elsewhere in the spindle. But these are the chromosomes with one stain, and this is the dynein. Our identification of dynein at kinetochores was a real breakthrough in a sense, because it's a motor that has the property that it walks towards the end of the microtubule that's way up at the pole. So the fact that dynein is at the kinetochore gave us a plausible hypothesis for how mitosis works. Dynein will bind the microtubules when they come along by chance. It'll hold onto them tight, and then it can walk on the surface of the microtubule, burning biological fuel, ATP molecules, to do it, and there's mitosis. Great. That's all there is to it. Well, guess what? For one thing, at about the same time, work was ongoing in yeasts to try to identify proteins that were important for chromosome segregation. And this kind of work goes on in yeasts much more quickly than in many other organisms, because yeasts are so wonderful in terms of being cells in which nobody minds if you kill 1 billion of them, for one thing. Every time you bake a loaf of bread, you commit genocide. But they also are convenient to manipulate genetically, and therefore you can make mutants of specific kinds. You can set up a screen to try to identify a phenotype, so-called. That is a characteristic that you want. Like, I want a yeast cell that segregates its chromosomes badly. Now that sounds like a terrible thing to do, but of course, if you find a mutant that does that, it's hard to maintain that strain, but you can do it, and it means that now you've messed up some gene product that is important for the function of the mitotic spindle. Working with that logic, people identified proteins in yeasts which were at first not understood, because although the sequence could be determined, nobody knew what it was. And yet what has gradually emerged is what they were finding in many of these loss of function mutations in mitosis was motor enzymes of the class kinesin, which is a completely different enzyme from dynein. And it turns out that every spindle uses multiple kinesins. And those kinesins drive all kinds of complicated processes. And this is now a slide showing speckles of fluorescence on the microtubules of the spindle. And this movie, taken by my colleague Ted Salmon, who's at North Carolina, and his then postdoc Claire Waterman, shows that the microtubules of the spindle are moving in both directions as if they were a conveyor belt. So the spindle, which I've shown you up until now as static entities, is actually dynamic in this quite remarkable way. And it turns out this remarkable flux of tubules towards both poles at once requires kinesins. Doesn't require dynein. It requires multiple different kinesins of different kinds. And indeed, as people have gotten more and more deeply into the motor enzymes that are involved in mitosis, it turns out there are many of them. I think it's 5 in budding yeast and 6 in fission yeast and about 14 in mammalian cells, as well as dynein at the kinetochores. What are these kinesins used for? Well, they bring about this flux, which I was just showing you in that movie, by bringing about the disassembly of microtubules at this end. There are motors that control microtubule dynamics and bring about microtubule disassembly. They're catalases that take microtubules apart from their ends. There are also molecules that promote the polymerization of tubulin. There are also motors that cross-link these microtubules that interdigitate here in the middle, and they're diagrammed here. And it turns out that there are kinesins that have the property that they push the microtubules in the green arrow direction, and kinesins that push the microtubules in the purple arrow direction. So this is very complicated because you have motor enzymes working oppositely against each other. And my interpretation of this is, you know, that if you want to do any operation that's a fine operation, like a spiccato up bow or something like that, you use opposing motors. The two fingers are working against one another to give you fine control. And I think that same principle is being incorporated here by cells at the molecular level to define a precisely controlled sliding of the microtubules, which is essential for the elongation of the spindle that I showed you in some of my earlier slides. So we have motors playing important roles, and having thought about motors for a long time and really sort of, in a sense, been the, apostle of motors in mitosis at a time when people really believed, no, it wasn't motors, it was just microtubule dynamics that moved the spindle. It was really very edifying for me to go through a period of the time when what my lab did was to obtain the very best evidence available that motors are not necessary for chromosome movement. And that was a shocker, because here I've been telling you how important motors are for building the spindle. What kind of evidence was it that led us in this direction? Well, the first evidence came from a system that was developed in my lab by Martine Couet as a postdoc and Vivian Lombillo as a graduate student. And we used a Tetrahymena cell, that's a ciliate, which we put in a blender and knocked off all its cilia and treated with detergent and washed off all the membranes. What we had left was just sort of a ghost of the cell. But it included all the structures that initiate the growth of cilia and flagella. And that could stick to a glass slide. And so we could now bring in tubulin that we purified from mammalian brains, and we could make natural microtubules growing out, which were highly dynamic. And then we could flow in chromosomes that we prepared for mammalian cells, and we could ask, what can we do that might make these chromosomes move? And here's a movie that shows some of Vivian's data. Down here is the Tetrahymena cell, and out here, you can't see them because this is not very good differential interference contrast phase, but that's a chromosome, and there's a chromosome, and I'm now going to flow in buffer from the right side of the cell, and you'll see the chromosomes move downstream as the buffer flow hits them, and the chromosomes are now under a viscous flow that is pulling them off to the left. But the buffer that's flowing through is a solution that contains no tubulin. So the microtubules now are going to dissolve because they are always in this dynamic equilibrium that you saw in that movie. And so as the tubulin falls off and the microtubules shorten, look what's happening to the chromosomes. They're moving at essentially physiological speeds. This is a little faster than physiology, actually, because this is not a time-lapse movie. It's just straight video. And what you're seeing is that simply the disassembly of tubulin can drive chromosome movement. And we were able to use a wonderful enzyme called a pyrase, which is a very promiscuous nucleotide hydrolase that'll knock down the concentrations of ATP and GTP to nanomolar levels, had no effect on this velocity. So this velocity is not driven by motors, it's driven by the disassembly of microtubules. So here was the disciple of motors all of a sudden saying, well, you don't need motors to move chromosomes. And indeed, a very good postdoc in my lab, Katerina Grischuk, who now is established in her own lab, did a wonderful experiment back in yeast. This was in fission yeast, in which she started with wild-type cells and set up an assay where she could measure the speed at which a chromosome would move to the pole. So this is simply the distance between a chromosome and the pole, and this is time. And you can see there's the speed of a normally moving chromosome. And now you can go ahead and you can knock out 1 motor enzyme, 2 motor enzymes, 3 motor enzymes. And we know, because this whole genome has been sequenced, this is all the motor enzymes that are in this cell, which could have the direction of motion of bringing chromosomes to the pole, and the speed at which the chromosome gets there is not affected by the removal of all the motors. Which says in yeast cells, in this fission yeast, you don't need motors for chromosome movement. You do need them to build the spindle, but once the spindle is built, you don't need them to segregate the chromosomes, and we were amazed. That result has been repeated in another lab on the same organism and in a different lab in budding yeast. So for the yeasts, motor enzymes are dispensable for the motion of chromosomes to the poles. Is that true in human cells? Very hard to say, almost certainly not. Because doing the right experiments in human cells is very difficult. I mentioned dynein. But if you take out dynein, you compromise a very large number of cellular functions. Dynein is a promiscuous enzyme used in multiple places. So nobody has the tool at this point which allows you simply to turn off dynein in a human cell. So I don't know how important dynein and other kinesins are for this motion if we were in a human cell. Humans are just lousy experimental animals, I'm sorry to tell you. But what we can do is we can study disassembly as a phenomenon and try to understand how much work can you get out of a disassembling microtubule. And would it be enough work to be able to account for the forces that are generated and the work that is done when chromosomes are segregating in a human cell? So that's the direction we've taken for a bunch of the work we've done. And this is now diagramming an aspect of the cycle of tubulin polymerization and depolymerization. This is a simulation done by my colleague Fazliya Tolakhanov based upon what's known about the geometry of microtubules as they assemble and disassemble. And this curling is a natural feature of tubulin when it has the nucleotide GDP bound to it, the diphosphate. When GTP is bound, these protofilaments are tending to be straighter and the microtubule will assemble. But intriguingly, when tubulin is assembling at some stage in this assembly process, which is pretty well understood but still under investigation, the GTP is hydrolyzed to GDP. So the microtubule is made of GDP tubulin. The funny thing about this is that if you take GDP tubulin and try to polymerize it, it won't do it. Even if you put in a seed, which is another microtubule that's stable, GDP tubulin will not assemble. So the interpretation of these observations is that if my two hands are now the two parts of the tubulin molecule, it has an alpha and a beta part. When you're GDP, you're bent. When you're GTP, you're straight, approximately. And when you're bent, you cannot assemble. But if you're straight, you can. But then when you're in a microtubule, if you hydrolyze, now if you think of one of these subunits here, it's surrounded by bonds. And even though it may be trying to bend, because it's GDP tubulin, it's constrained by its environment and it can't bend. But once you liberate its end, then whoosh, it can bend again. And so the concept that I give you as the model, which we've been in effect testing for a while, is that this bending that you saw is the relaxation of tubulin to its lower energy state. And if that's true, it can do work. Because if you're going from a high energy state to a low energy state, there's energy available, and so a disassembling microtubule could be a machine. And we ought then to be able to attach things to it, and in that way find out how much force can we generate. Well, we started out with a very simple experiment, and this was again work of Katya Grishchuk when she was a postdoc. And so we used the Tetrahymena pellicle again, and we used dynamic microtubules, and we took beads which we coated with streptavidin, and we used biotinylated tubulin. And for those of you not biologists, streptavidin is a protein that binds extremely tightly to a little tiny molecule called biotin. And you can attach biotin to some of your tubulin, and then the streptavidin biotin link will link that sphere onto the microtubule very tightly, and it is not a motor. It's one of the most static linkages known. And what we're going to ask then is, when these microtubules disassemble, will they make this bead jiggle? because if they make it move, the implication is that the disassembly process can make the bead move. Now, the problem we confronted experimentally is that if these were dynamic microtubules assembling and disassembling all the time, that means there's tubulin everywhere in solution. And so if we bring in a bead which is coated with avidin, with biotinylated tubulin, we saturate the surface of the bead with biotinylated tubulin before it reaches the microtubules. So we had to wash out all the tubulin before we could do that. But these are dynamic microtubules. You wash out the tubulin, they all disappear. So you have to stabilize them. So we put caps on them. And you can make a cap by means of using a nucleotide analog, GMP-CPP, where a carbon atom is in there. Now this won't hydrolyze. This tubulin is stable. So now we've got a stable cap. We can dilute all the tubulin away, bring in the biotinylated beads, and they stick. But we still have no experiment because we've got stable caps. But by using rhodamine tubulin, we had previously discovered that a fluorescent dye has the property that when you attach it to tubulin and you now stimulate its fluorescence, the microtubules will fall apart. And so what you can do is you now have made a photolabile stable cap on the tip of each of the microtubules, and you can get the experiment to go. You set the thing up, you start up a laser trap, which is going to come and trap the bead so we can measure its position and exert force on it. And now you go ahead and knock the cap off the microtubule. The microtubule is going to shorten, and we expect the bead to do a little jiggle. And the answer is, it does. The jiggle is pretty small. This jiggle is thermal noise, with the bead just dancing around as a result of Brownian movement. But this little dimple here is what we called signal. Not terribly impressive. However, we were able to show, if you think about this for a moment, this is a microtubule, it's 25 nanometers in diameter. The bead was a micrometer in diameter, which is 1,000 micrometers. So its radius means that its center would be way outside the room. We have a huge lever arm pulling on the bead, so that the force that we're observing way out at the center of the bead, which is where the trap is sampling the force, is going to be a tiny fraction of what any little fiber might do here. So if we went to smaller beads, we have less lever arm and the force should go up. 2 micrometers, 1 micrometer, half a micrometer, forces go up. So this is a credible result, although it's not an impressive result. And it's also not impressive because this is not a processive movement. This could never move a chromosome all the way to the pole. All it is a little jiggle. But on the other hand, cells don't use biotin avidin to couple chromosomes to microtubules. Is there a coupler that we could use that would be more physiological? And we were very fortunate because at the time this work was completed, there were 2 groups that were working on proteins that were involved in coupling yeast chromosomes to microtubules. And one of the most interesting and important one is a protein called the DAM complex. If you're from the East Coast, it's called the DASH complex. So you can take your pleasure whether you prefer to side with New York and Boston or with California. But we were collaborating with the group from the West Coast, so we call it the DAM complex. And it's actually quite frustrating to work on for a number of reasons. So calling it the DAM complex has additional virtues. This is what it looks like. Here's a microtubule in negative staining. There's the complex. It forms rings around microtubules, and each of the little subunits in this ring is 10 polypeptides. So this was really complicated. And the groups that worked it out had to identify all 10 genes, all 10 gene products, assemble them into a single bacterial cell that would express all 10 proteins in equal stoichiometry, and then they got soluble protein. And we collaborated with Georgiana Barnes, Dave Westerman, and particularly Stefan Westerman. Barnes is married to Dave Drubin, and their lab goes together, which is why I went to Dave. Stefan was the postdoc who did this work, and he sent us protein in our initial experiments, and then we started making our own. This is green fluorescent protein labeled DAM1 complex. And what we can do is we can use rhodominated microtubules, but we've got the green on there, and you're going to see a movie now in which you'll switch to the rhodamine channel, and there it is, and we're now going to knock its cap off with a laser. There it goes, and this is the way in which the dam complex behaves as the microtubules disassemble. It's a highly precessive movement. It's going over tens of micrometers, exactly what you would want for a chromosome coupling. So can we now couple this to our laser system? And we did this with antibodies to the dam complex. And under these circumstances, the forces that we measure go up by an average of a factor of about 5.5. Now that makes a lot of sense because our biotin avidin would have been right up against the microtubule, and it might have coupled to two protofilaments. There are 13 protofilaments in a microtubule, and with the ring sampling all the protofilaments as they bend, you'd expect about a factor of 5. So that was a very satisfying result. But the problem is this force is still only around 2 or 3 piconewtons, which is about the force that a motor enzyme developed, not quite as much. Piconewton is not a household word, I'm sorry, but this is a force measurement, and it turns out that myosin in muscle and dynein in flagella and kinesin all can develop around 5 or 6 piconewtons. And so we were a little bit below that, but we reasoned part of our problem here is if you look at this geometry, it's really weird. We're off on the side of the microtubule with this huge force disadvantage as a result of our lever arm. We want to be where a chromosome is, which is at the end of the microtubule. And in thinking about the coupling of chromosomes and microtubules, we realized that this is all not a very good model, but a better model for how to attach a kinetochore to a microtubule would be with strands of some kind. So here is a cartoon representation of the dam ring. Here is a bead, and what we're going to try to do is come around the way a kinetochore would be on the axis of the microtubule, and now we should experience the full direct force. So how do we make this? Well, we could have tried to do it by taking kinetochore proteins, but the trouble is then you don't know what's contributing what. So what we did instead was he took the long coiled coil part of a myosin molecule from cardiac myosin. And we had it because a colleague of mine, Leslie Leinwand, had it in her lab as a clone, and she had somebody working on it, and they had already made a version of it which was deprived of all of the cysteine residues that are naturally in that sequence, and he added one sequence right at the end, one cysteine. And then we took that molecule and we engineered onto this end of it a protein that binds extremely tightly to the green fluorescent protein. It's called green fluorescent protein binding protein, or GBP. And so we now had something we could attach to glass through the cysteine in a covalent bond and attach to the dam ring by means of the GBP and use fluorescent dam protein and see what we could get out of it. So now you're seeing a little movie, or you were. What's happened to it? Yes, okay. So these are graphs of what's going on. We've got a little initiator here in a microtubule and a bead, and it's attached, and we're going to watch as the microtubule comes along and its disassembly hits the bead. What we think is going to happen is that there will be a rearrangement in this situation as the force pulls. And the way we make the force pull is we use a spot follower circuit to be able to make this a force clamp, so that we're keeping constant force on this system and the stage is moving and we see how much force we get, and then we turn the stage off and ask how much force do we get when we pull the bead out of the trap. That was rather complicated. Maybe I should run that again because it went by too fast. Now, can I do it? Yes, there we go, it's running. Okay, so we're going to now turn on the trap and capture the bead that's attached to this microtubule. We're going to knock off the cap and let the microtubule disassemble. And as the disassembly comes, we're going to start exerting force. And we use a force clamp circuit now to move the stage so the force never gets any bigger. And we can exert something like 5 piconewtons on this. And during that time, some rearrangement occurs because the stage speeds up. And at this point, now we can turn off the force clamp so that we let it develop as much force as it possibly can, and that will then give us a signal of how much is the maximum force that the thing can develop. And these are the data. This is what the thing looks like, where the clamp goes on and we're allowing the rearrangement. Now we turn the clamp off and we accumulate force. And now instead of 2-3 piconewtons, we're now getting 30 piconewtons. So our prediction that we were losing as a result of the mechanical properties of our setup with the bead way off to the side seemed to be true. And what we've now shown is that a disassembling microtubule is 5 times more powerful than a motor enzyme. So it's like a bulldozer compared with, you know, the little Vespas that are sniggling around as a result of their kinesin activity. And we think that this argues that this is probably an important aspect of chromosome movement. This is probably enough force to do the kind of job that occasionally the spindle has to do. And I don't want to go into detail on this, but just moving a chromosome against viscous drag is easy because it's not a big problem. Chromosomes move very slowly, and therefore the viscous drag is low. But when you have chromosomes that are entangled and you want to pull them apart, you need to develop force. And a great experimental cell biologist, Bruce Nicholas, measured the amount of force that a spindle can generate. And although it's very hard to relate it to numbers of microtubules, it's up at around 30 piconewtons per microtubule. He estimated 45. But I think what this says is that we have here something that could be a serious player in chromosome movement. And it may be that for this fundamental motion of chromosomes to poles, which is sort of the most basic aspect of chromosome segregation, you don't need motors. Well, how does it work in mammalian cells? Because it turns out the irony is that this damn ring, which is such an elegant solution to sampling force from a microtubule, is found only in the fungi. So it's not a universal answer. And indeed, the way in which I think about this is that budding yeast in particular, it has only 1 microtubule per chromosome. And if you lose that microtubule, you're going to lose the chromosome, and that's a non-disjunction event, which is often lethal. So there's very strong selection in that kind of a cell for having a robust attachment that you're not going to lose trivially. Mammalian cells have an average of about 12 microtubules per chromosome. Plant cells, it goes up as much as 150 microtubules per chromosome. So many cells don't need this kind of device to secure chromosome movement, but how do they do it? Well, some components of the whole apparatus are conserved. Ajit Joglekar could tell you more about this than I can, but a protein like the so-called NDC-80 complex, which binds microtubules at one end and binds to the inner kinetochore at the other end, is conserved. But we made the discovery in trying to work with that protein that it could only sustain something like a half a piconewton or maybe 1 piconewton of force. It was a terrible force coupler. And we've tried various other things. So a recent discovery in human cells is the SCA complex. There are 3 proteins that assemble, they bind nicely to microtubules, they interact with the NDC-80 complex, and the SCA complex may well be, in some sense, an analog of the DAM1 complex, and yet we have the problem that with the SCA complex, again, you can't get big forces out of it. So is there anything you can get big forces out of? And the lead that I took was from sort of structural work done in my lab at a different time, looking at a flaring end of a microtubule. You know, you saw those bending protofilaments, and this is chromatin up here, and we found these little strands that run from the chromatin down to the bending part of the microtubule. And we thought maybe the secret is that the protein we're after is unknown because it isn't a conventional microtubule binding protein. A conventional microtubule binding protein would bind here on the wall, and that's where NDC-80 binds. But we're seeing something that looks as if maybe it binds perhaps to the inside of the microtubule, or perhaps preferentially to tubulin that's bent, of the kind you find at the end of the microtubule. So we set up an assay to try to find proteins that preferred to bind to bent tubulin. And you can do that. On the right are tubulin assembled into microtubules and stabilized with the drug Taxol. On the left is tubulin polymerized in the presence of vinblastin, which is a polymerase inhibitor, because it forces tubulin to form these little curls. And those curls have the same curvature as the curls at the ends of the disassembling microtubule. And so we took mammalian cells in culture, synchronized them in mitosis, made homogenates, and then did pull-downs. The trouble is that these pull-downs that we got with these two kinds of proteins are incredibly complex. And I was afraid we would never be able to sort this out. But there is this spectacular technology of mass spectrometry, which is now brought to such a fine art in a number of labs that you can take a complex mixture like this that contains thousands of proteins, and you can identify every one of them. So we collaborated with my chemistry collaborator, Natalie Ahn, who is very good at this. And with it, we were able to identify about 2,000 proteins in each of these different fractions. And what we could then do is use a technique called spectral counting, where you count the number of peptides that you get from each of the two versions of the same protein. So if I found myosin in one fraction, I would find myosin in the other fraction, and I'd compare the number of peptides that I got in my mass spec. Quantitative spectral counting it's called. And so you can go ahead and compare the relative strength of the two pull-downs. And here is a lot of data that you don't want, but if you compare the blue number here with the blue number there, you get a ratio which is that. And this ratio tells you the amount that this protein, talin, which is an actin-binding protein, prefers curls relative to taxol microtubules. Well, talin is not in the spindle, so although it was there, I wasn't that curious, but what I saw was centromere protein F that showed about a between 1.5 and 2.5 ratio of preferring to bind to curls. And centromere protein F has been a great enigma for quite a while. It's an extremely long protein, which has been identified by its founder, Tim Yen, as having a microtubule binding site at both ends. It's 3,114 amino acids long. It binds to kinetochores. It's known to be important in the kinetochore, but its significance is not well known because if you do a standard experiment of using RNA interference to block its expression, chromosomes don't attach properly to the spindle, but the kinetochore isn't built properly. You do not attach dynein, and you do not attach one of the essential kinetochore kinesins. And that means that you don't know whether the result you've seen is due to the absence of SEMP-F or the absence of the other things that can't assemble because SEMP-F is not there. So we need to dig deeper. And I don't have time to go into this in any detail, I'm afraid, but what we have shown is that SEMP-F binds very nicely to microtubules, and this is using Western blots in order to back up the mass spec data. And indeed, it prefers binding to GDP curls, which are a better example and model for disassembling microtubules. It likes GDP tubulin better than GTP tubulin. This long molecule has never been isolated, not by us or anybody else, but we've been able to use the techniques of molecular biology to express pieces of it using cDNAs that were sent to us very kindly by Tim Yen. And those pieces, diagrammed here from both the N-terminus and the C-terminus, have allowed us to identify properties of this molecule. And the intriguing thing, just to summarize, is that at the N-terminus, we have a site that prefers binding to the curls by a factor of about 4 to 1. At the C-terminus, we have a site that prefers binding to taxol microtubules by about 2 to 1. So these sites are different. And in our net effect that we got in the original mass spec identification, we were seeing the sum of the two bindings. But this molecule is attached to the kinetochore at a site here. And so both of these two sites are available to reach out and interact with microtubules. Does this play any functional role? And just again, quickly to summarize, what we now know is that the C-terminal piece binds well to microtubules, and it will diffuse along the surface of the microtubule quite freely. The N-terminal piece doesn't diffuse so well. Either piece can be attached to beads, And when they're attached to beads, we can do these force determination assays. And what we've discovered is that this protein is the best coupler known outside of the DAM complex. We're getting about 10 piconewtons of force with the beads that are coupled with this. And in a dilution assay, where we dilute the amount of protein on a bead, the C-terminal piece dilutes away quite quickly and we lose our force. But the N-terminal piece, we can dilute down, and we don't lose force until it all goes away. So it looks as if this may be an important aspect of the couplers that attach kinetochores to microtubules, how it functions in mitosis, how it works in association with dynein and other things that are important for chromosome movement are, of course, subjects for further study. But I think this is a good point in an Alan Hunt symposium to leave the issue, because as he said, we have more questions than answers, and this is a project which will last far beyond me as a scientific career. Thank you for your attention.
It's like an adventure that two guys have over a period of years, and so Alan and I have kind of been able to go together through this experience of myth busting in the microtubule world.